लाल बहादुर शास्त्री महाविद्यालय परतूर बायोलॉजी यूनिट एग्जाम्स मॉडल आंसर section A. Question number 1. Select and write the correct answer. The phosphodiester bond, tryptophan, bacteria and structure of DNA by Watson and Crick's model. Then question number 2, answer the following. What is point mutation? Answer: mutation due to change in a single base pair in a DNA sequence is called point mutation. Give the dual function of AUG. Next question, their answer: acts as initiation codon for protein synthesis. Second one is it codes for methionine. The next question is the state the function of non-histone protein. Answer is the packaging of chromatin. In one sentence answer, you must write in a one single statement is a must required to getting a full marks. Then section B, which one carry the each questions carry the 2 marks for their correct answers. Attempt any 4. Question number 3rd, the process of termination during transcription in a prokaryotic cell is being represented here. Name the label A, B, C and D. Now in this diagram showing the label A, B and C, you must write their answers what are the showing in the diagram. Now here A is showing the DNA molecules. Now B, mRNA transcript. Now C, RNA polymerase and D, Rho factor. Now the next question about question number 4. State the 4 criteria which a molecule must fulfill to act as genetic material. Answer: there are the 4 criteria you must write in the answers regards to the questions. It should be able to generate its replica, then should be chemically and structurally stable, should be able to express itself in the form of Mendelian characters. Then 4th one, should provide the scope for slow changes that are necessary for evolution. Next question is the question number 5. Give 2 reasons why both strands of DNA are not copied during DNA transcription. You must properly understand the question, what are the questions are framed, and to write the proper and orderly sequencing of that. If both strands code for RNA, 2 different RNA molecules, 2 different proteins would be formed. Hence genetic machinery would become complicated. Since the 2 RNA molecules would be complementary to each others, they would wind together to form dsRNA without carrying out translation, which means process of transcription would be fulfilled. Now section C, Attempt any two. Each question carry 3 marks. Question number 9. Give 6 points of differences between DNA and RNA in their structure, chemistry and function. DNA and RNA differentiation. Double-stranded DNA is double-stranded molecules, RNA is a single-stranded molecules. DNA thymine as a pyrimidine base, uracil is a pyrimidine base, pentose sugar is a deoxyribose. 2H makes it a reactive, quite stable and not very reactive, perform their function in the protein synthesis, dictates synthesis of polypeptides, perform their function in the protein synthesis. They are transported into the cytoplasm found in the nucleus. Question number 10. The base sequence on one strand of DNA is ATG, TCT, ATA. Give the base sequence of its complementary strand. Now this, what are the base sequence of the complementary strand? If an RNA strand is transcribed from this strand, what would you be base sequence of RNA? What holds this base pair together? Answer is there is a complementary base sequence is the TAC, AGA, TAT. Then after this, if RNA strand transcribed from this strand, what will be a base sequence of RNA? UAC, UGA, UAU. Hydrogen bonds, what will be these base pairs together? Hold this base pair together. Adenine and thymine are bounded by 2 hydrogen bonds, and cytosine and guanine are bounded by the 3 weak hydrogen bonds. Then question number 11. What are the 3 types of the RNA? Mention their role in a protein synthesis. mRNA, messenger RNA, decide the sequence of the amino acids. tRNA recognizes the codon on the mRNA and transport amino acid to the site of the protein synthesis. Then rRNA plays the structural and catalytic role during the translation. Then section D. Attempt any one of these below questions, given questions. Each question carries the 4 marks. Question number 12. What is meant by the semi-conservative replication? How did Michelson and Stahl prove it experimentally? Michelson and Stahl perform an experiment using the E. coli to prove that DNA replication is semi-conservative. They grow E. coli in a medium containing the 15NH ammonia, then C14, and then after the 15NHCl. Then after separated heavy DNA from this normal 14N by centrifugation in CS cell density gradient. DNA extracted after one generation of transfer from 15N medium to 14M medium had in an intermediate density. The DNA extracted after 2 generations consisted of equal amounts of light and hybrid DNA. They proved that DNA replicates in a semi-conservative manner. Then next one, what does the lag apeiron consist of? How is the operator switch turn on and off in an expression of gene in this operon? Explain. Lag operon consists of the following: structural genes ZYA, which transcribe a polycistronic mRNA. Gene Z codes for the beta-galactosidase. Gene Y codes for the permease. Gene A codes for the transacetylase. Promoter site where the RNA polymerase binds for the transcription. Operator acts as switch for the operon's repressor. It binds to the operator and prevent the RNA polymerase from the transcribing. Inducer in lactose is an inducer that inactivates and repressor by the binding to it, allows an access for the RNA polymerase to the structural gene and transcription. Question number 14. What is an operon? Describe the major steps involved in an operon. Operon is a group of controller and structural genes which control the catabolism of the cell and genetically lactose operant lack operons. Then when inducer or lactose is absent, the lac regulator gene synthesis a repressor protein by transcription and translation. This repressor protein binds with the operator site of a lac operon and blocks RNA polymerase. RNA polymerase unable to transcribe mRNA and structural genes, unable to translate enzyme beta galactosidase. When inducer or lactose is present, lac regulator gene transcribe mRNA and synthesis active lac repressor protein. At the same time, lactose is converted into isomer or lactose. All lactose binds to active lac repressor, due to which it is converted to inactive repressor. This inactive repressor is released from the operator site of lac operon and RNA polymerase binds to promoter and start to transcribe mRNA and forms beta galactosidase R, which convert lactose into the glucose and galactose. So the presence of lactose determines whether or not lac repressor is bound to the operator gene is expressed or not. So in this way, this in absence of the inducer, in presence of inducer, in presence of inducer and in absence of inducer, in this way there is the